Hello everyone. Today's topic will be proteins. I like to think of proteins as the doers of biology. And today we will be unpacking how they fold up, what kind of jobs they can do once folded, and how they do those jobs. We will be talking about proteins pretty much in every lecture moving forward in this course, so buckle on in. Today's learning goals will be to explain how amino acids underline protein structure and function. We will be identifying the forces that dictate protein folding, and then distinguishing the different roles of proteins. Today's molecule of the moment is a little different. It's not technically an actual molecule, but rather a theoretical one, a generic 100-mer polypeptide. Any 100 amino acids bound together in a single chain. And perhaps you remember from general biology that a protein is simply a polypeptide that is folded up in a specific, unique way that is critical for its function. Did you ever wonder how easy or difficult it is for the protein to fold, though? For a protein, 100 amino acids is chump change, so this should be easy. Let's make it even easier and say that each amino acid in the chain can try up to 2 conformations. Let's call it up and down if you want, or A and B. Knowing that, my question is, how many possible configurations then could this fictitious 100-mer protein fold into? And as a bonus, I want you to consider this. If we assume each possible folding event is 1 nanosecond long, what is the maximum amount of time for this protein to sample all possible conformations randomly? Work on that, and we'll discuss the answer soon enough. The answer to this molecule of the moment is perhaps the strangest one you've seen so far. This is actually a very famous thought experiment devised by a scientist named Cyrus Leventhal, and us biochemists call it the Leventhal's paradox. And we all know that a protein's fold is essential to its function. However, even one protein could wrap itself up into numerous possible configurations. Even small proteins just have too many degrees of freedom, it would seem, for them to fold randomly into the perfect knot every single time. How do proteins navigate the energetic landscape of folding? This diagram over here is called a folding funnel. And we use it to illustrate the many twists and turns and dead ends that a polypeptide needs to traverse in order to reach the stable sweet spot. And proteins find this minimum all on their own, without eyes or brains or Google Maps, almost every time. And they do it in less than a millisecond. I mean, how? How is it possible to go from a random noodle, essentially, into an ordered fold? There's a couple of reasons for this. It starts in part with the nature of the peptide bond itself. The sterics and electronics of the amide greatly restrict the number of allowable conformational possibilities. If we consider the skeletal structure of the amide bond as I've drawn here, it actually has a resonance form. There's a lone pair on this nitrogen. It can come down and kick up the pi system on the oxo moiety. resulting in a resonance structure that looks something like this. The CN bond over here, that, if you notice, as implied by the resonance structure, has 40% pi character. It's like a little bit of a double bond. And you may recall from organic chemistry that double bonds do not allow rotation. Hence, resonance of the amide bond reduces entropy. Let's highlight all of the bonds in this cartoon that are not rotatable. This CN, not going to rotate. This CN won't rotate either. Won't rotate, won't rotate. You may notice there are two very special bonds that can rotate. These are going to be sp3 hybridized, and they're going to be phi and psi, phi and psi, phi and psi. The absence of pi character in these phi and psi bonds allow for a degree of rotation. However, they aren't free to rotate willy-nilly. Let's take a look at this case over here. Let's draw out the polypeptide backbone when we set the phi and psi dihedral angles, they're called, to 0 degrees. If we take a look at the backbones, the oxo moiety of this residue and the amine moiety of this residue look a little uncomfortable. We have a steric clash. Two atoms can't be in the same place at the same time. So this is an example of a phi-psi dihedral criterion that is not allowable. Additionally, other angles that are uncomfortable for a peptide would be when phi equals 0 and psi equals 180, where we flip this residue 180 degrees. What's the problem with that? Again, we have the electron clouds associated with one oxo and the other oxo moiety of these two amide bonds clashing with each other, and that's not going to happen. The same problem happens in a different way if we were to flip phi instead by 180 degrees and kept psi at the original 0 degree criterion. When we do this, now we have the amine portion of the amide bonds from two adjacent residues clashing. And there's plenty other of phi-psi dihedral sets that are too high energy for a protein to fold into. The key takeaway here is that these unfavorable dihedral clashes are also a factor that reduces the initial entropy of the polypeptide chain. So I hope you're starting to realize that random noodle is really not a good description of an unfolded polypeptide. The second reason is the sequence of the protein itself. Like the DNA it comes from, each protein sequence is actually a code that dictates how the protein folds and the function it can carry out after doing so. When a protein enters solution upon synthesis, it immediately undergoes a process called hydrophobic collapse. shown over here. Take note of these green hydrophobic residues. Notice how they arrange themselves after doing this hydrophobic collapse. They bury themselves deep into the core of the protein to hide away from the water. Meanwhile, hydrophilic residues will extend outward and solubilize the protein's exterior. Polar and charged residues are more than happy to mingle with water. And after this initial partition, you will get then structural intermediates and fine-tuning, where the residues will optimize their side chain configurations and interlace together like puzzle pieces. Now, what does this process remind you of? If you're thinking the hydrophobic effect, you would be correct. The hydrophobic effect drives protein folding. Let's apply that concept to the here and now. We can first consider the process of going from an unfolded polypeptide to a folded one from the perspective of water. What's happening to the delta S, or the entropy, or the randomness of water through that event? Well, randomness for water will increase substantially. And that's because along this high surface area, low volume polypeptide, there will be plenty of ordered interfacial water, all hydrogen bound with each other. And lots of those waters are making, frankly, unfavorable contacts with those nonpolar side chains shown in green. When we undergo hydrophobic collapse, the surface area of that globule goes down, the volume goes up. With the surface area down, we shed those interfacial waters and we make them random. Hence the huge spike in entropy, and that favors spontaneity. What about delta H, the change in enthalpy? You can think of it as heat. Shedding those interfacial waters is actually endothermic because we're breaking the ordered hydrogen bonds all around the polypeptide. And that comes at an energetic price. Hence, delta H goes up. Heat flows in. However, the delta H term is much smaller than the randomness term. So if we were to compute delta G for this process, it would work out into the negative, which means spontaneous, and that's good. What about from the protein's perspective? Entropy here, randomness, is going down. We go from wet noodle to intricate knots. That does not favor spontaneity, right? Because the universe naturally proceeds towards disorder. The change in enthalpy also trends downward because what we're doing here upon folding is establishing intermolecular forces between the residues throughout the process, which is exothermic and hence favorable. But we're at a bit of a crossroads. Does the randomness term exceed the delta H term from the perspective of the protein? Well, the answer is not always, But the protein always folds spontaneously because of this coupling of water's release. The delta S term of water is so huge, it has entropy to spare, if you will, and that is what will drive the protein to fold, even if it winds up being an energetically uphill process with a positive delta G. Doesn't matter because water from the hydrophobic effect has our backs. By now, perhaps some of you are thinking, well, wait a minute. What about chaperones? Didn't we learn in general biology that the cell makes these giant containers called chaperones that will fold the proteins for us? That's actually a very big misconception. Chaperones are technically not necessary to fold the protein. It's worth repeating that the instructions to fold is the protein's primary structure. It's the sequence of amino acids, and that specific sequence is the folding manual. And a scientist by the name of Anfinsen did a really clever experiment to demonstrate this fact. What he showed in his seminal 1973 paper was he took an enzyme called ribonuclease, and ribonuclease is a bacterial enzyme that is held together in part by multiple disulfide linkages. And what Anfinsen did in the first half of his experiment was to unfold the protein. First, he cuts those disulfide bridges with a reducing agent. And then he added in an 8 molar solution of urea, which is a powerful protein unfolder. It dissolves all residues really well, and it essentially eliminates the hydrophobic effects. Therefore, we went from a 100% active ribonuclease enzyme to a 100% linearized or inactive enzyme. Anfinsen's goal now was to see if the protein could refold itself. So he split that sample into 2 separate pools, the experimental group and the control group. In both vials, he attempted to refold the protein in 2 steps, but he changed the order of operations. In the experimental case, he undid the damage by first removing that urea. This effectively turned the hydrophobic effect back on and let the protein scrunch up again. Second, he introduced air, which can reoxidize the cysteines back into their original disulfide bridges. Consequently, after doing this, Anfinsen restored 90% of the ribonuclease's functionality. And I'll remind you, all of these experiments were done in a test tube. No cells or chaperones were around to guide the protein back together again. Now we can consider the control group. The same thing, but Anfinsen first oxidized the cysteines in air. However, the protein this time was linearized and random though, so most of the cysteines didn't relink in the original way. Then Anfinsen finally removed the urea and the hydrophobic effect kicked in, but most of the protein was already permanently distorted by the scrambling of these disulfides, so it was stapled together in the wrong way. So Anfinsen was only able to restore 1% of the original activity compared to 90% in the experimental group. Thanks to this famous experiment, we can see how the hydrophobic collapse of the primary sequence alone is all we need, technically, to get the fold right. To recap, there are two reasons why the protein folding problem isn't nearly as impossible as it seemed in our molecule of the moment. First, the backbone of any protein is chiral, stiff, and has restricted phi and psi angles. The second reason is that the primary structure dictates folding. That's nice. Well, what is the primary structure, the sequence of amino acids, folding towards? If the specific order of said amino acids is the protein's primary structure, then the secondary structure, that consists of regional folds, which are established and held together through backbone hydrogen bonding interactions. That is what the primary structure builds towards first. And there are two very common secondary structural elements called the alpha helix and the beta sheets. Let's first discuss alpha helices. These are the most common secondary structural elements. About 33% of all protein secondary structure adopts the alpha helical arrangements. And you can spot them pretty easily when the protein backbone coils about this invisible axis in a right-handed manner, just like so. I also want you to take note of the positioning of the R groups, which in this diagram are shown as purple balls. There, 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 and there. Notice how they all point outwards. Maybe you can see that more clearly in this diagram over here, where we stare down the barrel of the alpha helix. Notice how the side chains all extend orthogonal from the elongation axis. The side chains protrude outwards. That's a really important thing to know. A lot of students, I think, tend to think that those R groups are buried within the helix, within the interior, kind of like DNA, but there's just not enough room in there for that to happen, they point out. Additionally, perhaps you can see that there's plenty of backbone hydrogen bonding going on along the helices axis. We can visualize these as the dashed blue lines shown over here, for example, and here, and there, and all over the place. Specifically, these are hydrogen bonding interactions between different backbone amides on adjacent rungs. And I'll try to draw that over here in a molecular way, where the amine moiety of one backbone group can hydrogen bond with the oxo moiety of another that's downstream, like that. This extensive hydrogen bonding throughout the coiled backbone gives the alpha helix rather reliable structure, And here are some important geometric values that you should know for alpha helices. First, there are 3.6 residues per helical turn. You can get a sense of that non-integer value in this spiral diagram over here. Let's count the amino acid number as a function of spiral position. If you go 1, 2, 3, 4, that's approximately one turn. An additional 1, 2, 3 roughly constitutes another, and it sort of bounces between 3 residues in a turn, 4 residues in a turn, so it averages out to 3.6 residues per turn. Additionally, each turn rises by 5.4 angstroms. You can check out this diagram over here. It got a little bit messy, but the distance between this rung and this rung dials in on average to 5.4 angstroms, and there would be 3.6 residues on average in between here and there. And I should probably remind you that an angstrom is a very, very small distance measurement, where 1 angstrom equals 0.1 nanometers. It's about the width of a single hydrogen atom, by the way. Lastly, be aware that an alpha helix is typically 4 to 40 residues long, and that averages out to 10 per helix, give or take. Here's the tricky part. I keep telling you that the primary structure is what dictates the higher levels of folding. So you may ask which amino acids specifically code for building alpha helices. And the answer to that question is MALEC. That stands for methionine, alanine, leucine, glutamate, and lysine. I hope you're studying those amino acids, single letter codes, structures, and yada, yada, yada. What helps me remember this is the actor Rami Malek. You probably, well, I know him from the TV show Mr. Robot. Perhaps you've seen him in other things since then. Either way, his last name Malek just happens to have all the single letter codes for the amino acids that build alpha helices, the so-called helix builders. And something geometrically that the helix builders have in common is that their R chains tend to be very linear and slender. That makes them very compatible with helical structures because their side chains can stick outwards with minimal clashes, like so. I'll just draw a few squiggles to show how they just point out and don't crash into each other. And furthermore, look at how they line up every 3 to 4 residues or every twist of the alpha helix. Let's imagine, for example, that this residue was a positively charged lysine, and this residue was a negatively charged glutamate. Look at that. There's a favorable interaction now that would further stabilize the alpha helix. Hence, when you examine a protein segment, look for the sequence malic or any combination of those letters. It may not necessarily have all of those in that order. It could just be a bunch of alanines in a row. But what I also want you to do is look for any of those malic residues with an I plus 3, an I plus 4, or an I plus 7 registry, it's called, or a spacing of every 3 to 4 and such. Especially, in this case, where we have a lysine and a glutamate that are separated by 3 or 4. Now let's talk about the opposite, the helix breakers. What are the helix breakers? They are going to be the residues proline and glycine, and the reason why can be extracted from their molecular structures. Glycine is the simplest amino acid. You can think of it as lacking an R group. It's just another hydrogen atom, and the consequences of that is that glycine can rotate and twist its phi-psi angles really easily. And as a result, it's considered to be a very floppy or fluxional amino acid. It has too much entropy, essentially. And what that will do is create a weak point along the helix from which the entire thing can unravel. On the flip side, proline, which looks like this, that's a very strange amino acid because its R group wraps around and binds to the N terminus. And that makes it very restricted and sort of forces it into very stiff, strange phi-psi angles when it is bound in a polypeptide sequence. That kinkiness, that distortion, doesn't align well with the natural curvature of the alpha helix. So its kinky nature also makes it a helix breaker. As a matter of fact, the residues proline and glycine, we often find them at the ends of alpha helices. And in a sense, they kind of serve like instructions to the molecule that this is where the helix ends. And that's because glycine is too loosey-goosey and proline is too inflexible. The second most common secondary structural element is the beta sheets. About 20% of all secondary structures look like this. These will comprise the more linear segments of a polypeptide. Notice how they look like arrows rather than coils. I also want you to take note of the R group positioning. In this diagram, we will still represent the R groups as purple balls, and notice how it points up in one residue, and it's hard to see, but they point down beneath the plane of the arrow in the next one, and we alternate pointing up, pointing down, above, below, and you may notice how that causes the texture of the beta sheet to approximate this accordion-like ruffling, which we call pleats. Hence, why beta sheets are sometimes called beta pleated sheets. Unlike alpha helices, which prefer slender amino acids, beta sheets love bulky and branched residues. Think beta, bulky, and branched. Lots of B's. Some examples of the bulky amino acids would be F for phenylalanine, W for tryptophan, and Y for tyrosine. If you need fun little tricks for memorizing these, phenylalanine starts with the F sound. When I think of tryptophan, I think tryptophan. And tyrosine, I like to think of it as tyrosine. That's how I say it in my brain. So maybe that'll help you memorize FWY. These are the aromatic amino acids, the hydrophobic aromatic ones, and they're all going to be quite bulky. The branched amino acids, vilt, valine, isoleucine, leucine, and threonine, these are going to go very nicely within a beta sheet. And you may be thinking, hey, isn't leucine part of malic? And the answer is yes. That's why predicting these secondary structures can be very difficult. Sometimes the rules overlap a little bit. Leucine is slender, but it has a branch in it for sure. One of the other reasons why leucine can moonlight between beta sheets and alpha helices is that beta sheets in general tend to be very hydrophobic. All of these residues are nonpolar. Let's talk about their geometries. On average, they will contain 6 residues within every strand, give or take. And in general, you will find less than 6 beta sheets per protein. The beta sheet gets too long or you get too many beta sheets, bad things will happen, let's just say. Additionally, let's talk about the interplete distance. The interplete distance is roughly 6.5 to 7 angstroms. And you can see what the interplete distance is in the corresponding cartoons. Think of it as the distance between either valley to valley or peak to peak within these pleats, so to speak. You may also remember from general biology that beta sheets exist as 2 subvarieties. The first is called the anti-parallel arrangement. This is the more stable, more common beta sheet variants where the segments run in opposite directions. You can see that in figure B. And the anti-parallel arrangement is common because proteins can easily zigzag like this when they fold. The sequence can easily meander, just continue with a turn over there and go back and forth, back and forth. This arrangement likes to form when bulky or hydrophobic residues alternate in every other position, which is referred to as I+2 registry. All right, think about it. The R groups in beta sheets alternate up, down, up, down. So the residues that are spaced I+2, these are the ones that will be interacting with each other because they'll be on the same side of the sheets. Another way of phrasing this is that we get anti-parallel beta sheets when we exhibit something called monofacial hydrophobicity. When we have a hydrophobic face and a hydrophilic face, for example, on the other side. And again, these prefer hydrophobes and bulky amino acids with I+2 spacings, but particularly the bulky ones prefers a bulky I+2 arrangement. Parallel beta sheets are a little harder to form. These are less common and less stable. And you can sort of think this through. First of all, notice the hydrogen bonding interactions between the parallel strands. See how they're a little bit longer and angled and uncomfortable looking? The hydrogen bonds in the anti-parallel case, they're shorter, they're more direct. It just looks like it fits together more neatly, right? Secondly, I want you to think about entropy. In the case of the anti-parallel beta sheet, it's really easy to wind back and forth, like so, with very short, simple turns. In a parallel case, you may have to have these really long segments that loop around and backtrack. And that seems to have a higher entropic hill to cross to stick together in this parallel form. In terms of primary structure, what usually favors the formation of parallel sheets is bifacial hydrophobicity. I'm not saying we should see a phenylalanine every other residue, like in the case of I+2 registry. I'm saying a bunch of phenylalanines and valines and tyrosines and threonines all grouped together, like I+1 registry, if you will. That way, we'll have hydrophobes on both sides of the pleats, and that's something we commonly observe in parallel beta sheet geometries. Additionally, there is a slight preference for extended glutamines and asparagines as well. And a trick for glutamine, I like to go glutamine-cutamine. And the reason why that's very common is because the glutamine side chain, come on, are really good hydrogen bonders, and the hydrogen bonds can stabilize the side chains in that manner, which can promote beta sheets big time. More so in the parallel case, of course. I also want to quickly add on that in addition to beta sheets, sometimes you will find these clipped off with a specific sequence called the beta turn. And the code for that is XPGX, where any residue and any residue are punctuated by proline and glycine. And you can structurally envision perhaps why this will allow sharp turns to exist, particularly in the case of anti-parallel beta sheets that zigzag in this accordion-like fashion, or this meandering-like fashion, because the proline establishes a hard turn, and the glycine gives us the flexibility to bend a little bit more and re-enter the next strands of the beta sheet. So I hope you're beginning to appreciate how the different personalities of the amino acids lend themselves to certain kinds of structures. I think this is pretty cool. A scientist by the name Ramachandran got a very clever idea. He figured since most proteins form alpha helices and beta sheets, and that these motifs can only form from certain backbone configurations, is there a way to correlate the sum of the phi and psi angles to the protein's secondary structural elements. What's the relationship there? Well, what he did was he would look at crystal structures of proteins and plot out the phi and psi angles between each residue along the primary sequence. And he would plot them like so. He would write down the phi angle as an x-coordinate, and the psi angle would be recorded as a y-coordinate. So for the first phi-psi pair, you would put a point on the plot. The second one may be somewhere completely different. And he would do this all throughout the protein. And he would do this for many proteins. And he noticed patterns. Scientists love patterns. The pattern being clusters of angles tended to correlate with specific secondary structures. Take a look at this table. For example, he noticed on average, alpha helices tended to have phi and psi angles that were around -57 and -47 degrees respectively. If you look at this plot, -57 is, I don't know, maybe like around here. -47 is around there. Where do they meet? Right in the island of alpha helical angles. And you can do this with any number of secondary structural elements, anti-parallel beta sheets, parallel, so on and so forth. This is a really handy tool for getting the secondary structural blueprints of a protein, wouldn't you agree? And again, I think the implications are even bigger than that. I think it really helps you appreciate how much entropic space nature has learned to disregard simply by the design of the protein backbone. What do I mean by that? Hardly any phi-psi angles exist in this quadrant. And this quadrant is also practically empty too. So the inherent stiffness of the backbone that we talked about earlier in this lecture, we can see physical evidence of it over here as an absence of large swaths of phi and psi angles. The final secondary structural element is a bit of a misnomer. Random coils are technically the absence of secondary structure, but that does not mean they are quote-unquote random. These are stretches of residues between more ordered domains, such as alpha helices and beta sheets, and you can also find random coils at the N and C termini of a protein, which are sometimes called the terminal arms. That's because they tend to be more exposed and floppy. Here is an example of a random coil segment. That's the C terminal arm of ubiquitin. And you can see random coils connecting these beta sheets and bridging this beta sheet and that alpha helix, so on and so forth. In general, we could say that hydrophilic residues tend to be disorder promoting. That's not always the case. The residues that are most commonly found in random coil segments with increasing frequency are alanine, glycine, arginine, aspartate, histidine, glutamine, lysine, serine, glutamate, and proline. You may notice that there are some contradictions though. Alanine is not hydrophilic, it's hydrophobic. Glycine is kind of on the fence, to be honest. We also learned that alanine, lysine, and glutamate are going to be helix builders, not random coilers. Proline is also a good example of a hydrophobe. And what else? Glutamine. That's a parallel beta sheet builder, not a random coiler. So I'm pointing out these contradictions for a reason, in part to make your lives difficult, I suppose, but also to kind of demonstrate that predicting protein folds is not as of an exact science as you would probably initially believe. But make no mistake, nature always knows what it's doing, more or less. I'll direct your attention back to the ribbon diagram of ubiquitin. Sometimes it's affectionately called the protein killer. If a protein has degraded or denatured in some way and the cell has no more use for it, it will often tag it for destruction and try to recycle or excrete its components. And it does that by sending ubiquitin after the protein, which will literally tag it for destruction. And it does that thanks to its very disordered C-terminal region, RLRGG, underlined and bolded over here. Disorder is beneficial in ubiquitin's function in the C-terminal region because this segment is kind of reactive and it can worm into almost any protein pocket, grab on, and flag the executioner to come over with its axe. So we often talk about how the structure and the fold of a protein is essential, but not every part of every protein is going to be ordered. Sometimes we need a little bit of randomness to get the job done. The next level of protein structure is tertiary structure. These consist mainly of local folds that are stabilized through side-chain interactions, which is distinct from the backbone-backbone hydrogen bonding observed in secondary structure. What I want you to do is pause the video and you tell me which amino acid side chains satisfy the following interactions. We can start with the hydrophobic and pi-pi interactions. And there's an example in this diagram to the right, shown in that magenta box. I'm noticing two valines. Notice how the valine side group looks like a V. V for valine. They're both interacting in the core of that protein with what looks like London dispersion forces. And valine's a good example of a nonpolar amino acid, so all of the nonpolars are fair game here, which we discussed last lecture. Pi-pi interactions you may or may not be familiar with. In case you're not, I'll tell you now that this is a favorable interaction between two aromatic systems, and it's sometimes called pi stacking. Those are like two benzene rings stacked together. So which side chains have aromaticity to them? That would be the bulky FWY, phenylalanine, tryptophan, and tyrosine. Nice. The next example are the hydrogen bonders. Which amino acids are capable of doing this? Shown here in blue. We're given two examples off the bat. That top residue looks like a serine to me. It's got a hydroxyl. And the bottom residue, that's kind of interesting. It is an aspartate, but this oxo group can certainly be a hydrogen bond receiver, just like so. What are some other residues that can participate in hydrogen bonding? Serine's cousin threonine, aspartate's cousin asparagine. We can't forget glutamine, the Q-tamine for Q. What else is there? Tryptophan, believe it or not, has a nitrogen, which means it can be a hydrogen bond donor. Tyrosine can be a hydrogen bond donor or receiver, even though both of those are hydrophobic. You can still hydrogen bond. And histidine. It's sometimes a base, sometimes acidic, but the nitrogens can go both ways in its case. Salt bridges are up next. In this diagram, we see an ionic bond, which can be synonymous with a salt bridge, between an aspartate again, and, well, this looks like it was meant to be a lysine. They dropped a few methylene groups in between, but we will assume that it's buried within this part of the purple line. Essentially, the residues that can do salt bridges will be the acids and bases. So D and E, aspartate and glutamate, will be your acidic amino acids, K for lysine, H for histidine, and R for arginine. That will be the basic ones. Acidic, basic. And this may be the easiest or hardest one to solve. The disulfide bridges, there's only one that can do it. Cysteine. Cysteine has a free thiol that can oxidize into this SS bond. It is the only covalent tertiary interaction. The last kind of protein structure is called quaternary structure. And these are the interactions that arrange multiple polypeptides into a single protein. And in my opinion, quaternary structure is the most confusing. I'll remind you that the definition of a polypeptide is any one chain of amino acids, whether or not it's functional. A protein is one or more folded and functional polypeptides. So a polypeptide can be a protein, or a protein can consist of multiple polypeptides. I like to consider quaternary structure, therefore, to be the optional kind of structure, because all proteins contain primary, secondary, and tertiary structure, but not all have quaternary structure. There are plenty that are happy to be just a single chain. How do these polypeptides come together? It's pretty much driven by hydrophobic interactions. Very often there'll be hydrophobic surfaces on one chain and another chain, and they will structurally complement each other and click in place, again, like puzzle pieces. However, salt bridges and hydrogen bonding contacts, and even disulfides, pretty much any kind of tertiary interaction can be employed between polypeptides to stabilize the quaternary structure. So it's not just any given one thing. It's often a combination of these working together. Quaternary structure also has several subvarieties. The simplest kind of quaternary structure is called isologous. That is when you have identical chains, identical subunits, experiencing identical contacts, like your hands clapping together. Isologous quaternary structure often forms simple dimers, and an example of that is alcohol dehydrogenase. This is the enzyme that detoxifies alcohol. And you can sort of see in this picture, got a whole mess of alpha helices and beta sheets, but there's this symmetry between two identical protein subunits, there and there. The same chain, just glued together, head to head. Next up, contacts very often can be non-reciprocal. And when they are non-reciprocal, that's like a hand stacking on top of another hand, all right? We call that a heterologous association, and that can open the door for more complex quaternary structures. One example of a heterologous association is observed in hemoglobin, where we have two alpha subunits and two beta subunits combine together to form this dimer of dimers in tetrahedral symmetry. Actin is a crazy example of a heterologous association. It's a single monomer that stacks up head to tail, so heterologously, and that allows it to stack along almost indefinitely and form these gigantic filaments. Tubulin is an even crazier example because that contains a heterologous dimer of an alpha unit and a beta subunit, and then those will have an additional dimension of quaternary structure that wraps around with this helical or translational symmetry, and it can form these zillionomers of quaternary structural elements. And technically, entire variants are a type of quaternary structure as well. This tomato bushy stunt virus is a sextamer of heterotrimers all arranged in radial or spherical symmetry. See the patterns of the colors? That's technically one protein, the entire capsid, which is formed from the individual capsid units. At this point, I'd like to finally address chaperonins. Remember that folding funnel metaphor? How a protein must navigate structural intermediates en route to its proper fold? Well, the native state, the correct fold, isn't necessarily at the bottom of the folding funnel, which is to say the thermodynamic minimum. As a matter of fact, it's often almost never the lowest energy, most stable configuration. The bottom of the well is not a good place to be. It's sort of like a black hole where proteins fall in and they go to die. And the name of that well is called amyloid. Amyloids are misfolded proteins. They're misfolded in a way that's too stable. So stable that when other proteins bump into it, the amyloid corrupts it and zombifies it and turns it into one of its own, and the fibril will grow and get bigger and bump into more proteins that will corrupt more and more proper folds in the cell. It's kind of like a protein graveyard. And these amyloid fibrils that grow can be visualized via electron microscopy, which you can see over here. It's very beautiful to look at, but they're very bad for the cell. You may also notice that there's a lot of beta sheets in these amyloid fibrils. That's why we want to keep the beta sheets in a protein often to a minimum. They're a little bit too stable. And as a result, amyloids are often considered to be toxic, and these amyloids are going to be part of Alzheimer's disease and Parkinson's disease, mad cow disease. All these neurodegenerative disorders stem from protein misfolding, according to most theories. And in a healthy cell, chaperonins will protect against this. They will be standing outside that amyloid graveyard, and they will help push proteins away and send them to the right direction before they get killed. Chaperonins come in 3 different classes. The first is called a holdase. I'd like to think of this as timeout. A holdase is a compartment where amyloid proteins can be sent and fold up with themselves properly before they become the seeds of these amyloid fibrils. Okay, not every protein needs a foldase. I believe less than 10% require them. But this is a passive form of assistance. No ATP required. Foldases do require ATP. These are very expensive, and we only whip them out when proteins get partially misfolded and they start creeping towards the amyloid well. What the cell can do is grab an exposed hydrophobic portion of the misfolded protein, reel it into the foldase, and whack it against the walls, kind of like when you take clothes out of the washer or dryer and you sort of just like whip them to get the wrinkles out. That's the exact same methodology, basically. And that lets you restart and try folding again properly. The third kinds are called heat shock proteins. These chaperonins are only made when the cell is very hot or very cold, which threatens the folding process. And these also require ATP. And so we can help all the proteins when we reach extreme environments. Protein structure is as intricate as it is crucial to its overall functionality. And proteins are not immortal. If you take that higher order structure away, you are going to lose the functionality that the fold instills. And proteins can lose their structure or denature in many different ways. Let's consider a very common protein that you have likely encountered in your day-to-day lives, serum albumin. The function of this protein is to regulate osmotic pressure, among other things. It's a good water binder, and you find a lot of albumin in egg yolks, for example. That's what is responsible for the gelatinous, slimy texture of egg whites. So what happens, let's say, if you were to cook egg whites, like hard boiling an egg? You're going to change the properties of that protein. It's going to go from a liquid goo to a hard, firm gel-like consistency. So heating a protein is one way that you can denature it, aka cooking. This, by the way, is why cooking food sanitizes it, because we are denaturing the toxic proteins that are in any foodborne pathogens lying in that food substance. Now, what's another way that we can destroy serum albumin? If you were to heavily whisk the egg whites and incorporate air into it, you're going to turn from a viscous slime into a light and airy foam, aka meringue. So mechanical agitation is another way that you can disrupt a protein's higher order structure. Secondary, tertiary, quaternary structure is not held together very tightly. So just quickly moving it or shaking it is enough to often destroy a protein's functionality. Other ways that you can mess up a protein is through the addition of organic solvents. So let's say we add alcohol sample that contains protein. That can cause it to instantly precipitate and coagulate, because what are we doing to the solution environment? We're changing its polarity and dielectric. We're namely reducing it. And that's going to ultimately screw up the hydrophobic effect. The nonpolar regions of those proteins are going to be less inclined to stay buried. They may present themselves outside, which is going to kind of turn the protein inside out. You can sort of think of it that way, which is going to screw up its higher order structure. Another way of messing up proteins is adding in acid or base. By changing the solution pH drastically, if there are acidic or basic residues in that protein, it's going to potentially change their protonation state, which can interrupt certain tertiary or quaternary interactions, such as salt bridges, that would normally stabilize the native state. And then also, to a lesser extent, salinity. Adding in a bunch of salts is going to change the ionic potential of the solution, and that's going to mess with the pKa's in particular of acidic and basic side chain residues, which can also screw up your salt bridges. So there you have it. Heat, mechanical agitation, organic solvents, acids and bases, and high concentration salt solutions can all interfere with the weak interactions that hold the proteins fold together, namely secondary structure or greater. I also want to mention that we can actually utilize protein denaturation as a way of assessing how robust a protein is. Check out this plot down over here. What we are doing in this experiment is comparing the stability of two different proteins, ribonuclease A in the blue curve and apomyoglobin in the red curve. And what we are doing in this experiment is increasing the temperature of the solution. And while we do that, we're going to track the percent of the protein that unfolds or becomes denatured. And the first thing I want you to notice is that it is at first gradual and then drastic process denaturation. It has this roughly sigmoidal function to it. It's not all together or all apart. The changes are, to varying degrees, gradual. And not only that, if we were to identify the inflection point of this curve, the Tm, the melting temperature, we're able to note that these proteins become 50% denatured at different temperatures. For ribonuclease A, it looks like it's denaturing around, 55 degrees Celsius, whereas apomyoglobin reaches that 50% denaturation mark closer to 65 degrees Celsius. So what we can interpret from this graph is that apomyoglobin is considerably more heat stable than ribonuclease A. And the cool thing is you can change the variable in the x-axis to be any of these 5 different denaturants. Instead of looking at the sensitivity to heat, you can try changing the x-axis to pH or percent ethanol or percent sodium chloride as a way of determining how stable your proteins are to each of these different denaturants. Let's now discuss collagen. This is a weird, exceptional case to much of what we've discussed so far, but it's also 30% of all the protein in the body by dry weight. It's the main structural component to connective tissue, so it's pretty important to understand this. Collagen features a very special structural motif known as the triple helix. Its primary structure consists of these glycine, proline, hydroxyproline repeats, and when you take these 3 amino acid residues and iterate them in a polymer, it's going to tend to adopt this left-handed helix orientation as a single polypeptide. But collagen doesn't just exist as a single polypeptide. these left-handed helices actually stick together in these triple helical bundles, which you can see over here, and they're going to arrange like so in this repeating pattern. And they're going to coil about and extend lengthwise to very, very long regimes. Now, you may be wondering, hydroxyproline, that doesn't sound like one of the normal amino acids. And you're right, this isn't a natural amino acid, hydroxyproline. Technically, it's called 4-hydroxyproline. And you can see an image of it over here. In the fourth position, we have inserted this hydroxyl group. And we do this post-translationally with a vitamin C dependent enzyme. And that's what imparts the hydroxyl on the proline, which is going to let it do extensive hydrogen bonding between the helices. You can get a feel for that over here. We're going to have these hydrogen bonding networks that will feature backbone-backbone hydrogen bonds, as well as hydrogen bonds between backbone and side chain, and even structural waters, like kind of got buried in there, but all of this is important for gluing the collagen fibrils together. So what happens if our diets are not rich in vitamin C? Let's say we don't have a lot of fruits and vegetables all of a sudden. When we don't have vitamin C in our diets, that's going to result in aberrations in collagen's macromolecular structure. The word for this disease, you may know, is scurvy. Now, what are the symptoms of scurvy? These are some illustrations taken from the Journal of a Naval Surgeon. Sailors at sea did not have access to abundant fruits and vegetables, so this was known as the sailor's disease back in the day. And what would happen to these sailors? Their teeth would fall out. They would have skin lesions. And there would be tons of bleeding and poor wound healing. And that's because the body has lost its ability to make effective collagen. If we don't have vitamin C, then we don't get any hydroxyproline. And there goes tons of our critical hydrogen bonders needed to stabilize the triple helix. So the collagen falls apart, which means your connective tissue and your skin falls apart. And that, I think, is a great example on how lots of disease pathologies ultimately trickle down to the molecular level. Hence, form befits function. So this last slide is really more bonus material, if anything, but we did spend a lot of time talking about the rules of protein folding. So you may be wondering, how easy is it to predict protein structure from primary sequence alone? Said another way, how good are we at designing proteins from scratch? And truth be told, we're still not amazing at it. There's a scientist at UC Berkeley, his name is David Baker, and he's famous for making one of the first designer proteins, like this one over here, which is called Top 7. It has no function, but it shows that he built a program called Rosetta at Home that was able to build and correctly guess this exact protein structure just from the sequence alone. And this happened in 2003. Just so you know, that's the year that iTunes was made. That's 2 years after the Nintendo GameCube. That's how long it took from the discovery of proteins to us generating the ability to think one up and actually have it fold the way we intended.
And what's really amazing is that he published his algorithm, this Rosetta at home, and his lab actually helped bake it into an online video game platform called Foldit. Like you can Google this right now and play the game. This here is a screenshot of the Foldit interface. And what Foldit is, it's a puzzle game. It gives you a cartoon protein and it tells you how to pack it together like a puzzle. It doesn't tell you anything about the hydrophobic effect or what a glycine is. It just says move the pieces to fit together so they just assemble like Legos. And meanwhile, there's an AI running in the background that's studying your inputs and learning, and it's using your knowledge to refine Rosetta. And thanks to tens of thousands of frankly literal middle schoolers just having fun on this video game, these scientists were able to redesign a protein to catalyze a reaction never done before by biology. It invented a Diels alderase. And these results were published in a very respectable journal in 2012. So, this is something that I say time and again with biochemistry research. The sky is the limit.